안녕하십니까?
Good morning.
BIOPLUS-INTERPHEX 코리아 2022
This is a BIOPLUS-INTERPHEX 2022, and my name is Bae-shin Kyu. I will serve as a moderator for this
이번
session.
행사는 3년 만에
After 3 years,
치르게 돼서
we are holding this event offline, so it is very meaningful. And despite your busy schedules,
귀한 시간 내주신
you took your valuable time. Thank you very much for your presence. Then we would like to begin the session now.
엑소존 기술은 최근
The exosome technology
바이오약품
is an innovative technology, and it is getting attention for biopharmaceutical drug development. And as a platform, it has the expandability, so it is getting attention around the world. And now we have the pioneering head of the companies developing exosome technologies to listen to their exosome experience. And we would like to learn about the scalability, and I believe we can also learn about the trend of the technology. In this session, we have 5 speakers, and we also have a 20-minute panel discussion. Then I will move to my chair, and I would like to introduce the first speaker, and we will begin the session. And while I introduce the presenter, the speaker, please come up to the stage.
첫 번째 발표하실
So the first speaker
발표 제목은
will talk about the adipose stem cell exosome for regenerative therapeutics and aesthetics. And Mr. Jo Hyung-sung established Exosome Bio in 2017 as a CEO and CTO. He received the master's degree and doctor's degree in Seoul National University and also received the business administration master's degree in KAIST. And he also received the investment from venture capital, and he also has an experience as an investor. Then I would like to ask you to begin the presentation. Good morning. Thank you very much for this good opportunity. The topic I would like to talk now is exosome, which I focus on, and I also would like to talk about the business as well.
엑소코바이오는 작년에
Exosome
25개 글로벌 유망 헬스케어 기업과 함께
along with the 25 prominent companies, was covered at BBC. BBC selected 25 companies, and among them,
함께 그 제목에 보시는
along with Exosome Bio,
엑소솜의 본연의 기능이 메시지 전달에 관련돼서 저희가 특별 프로그램을 만든 적이 있습니다.
we created a special program, as you can see in the title, and this program is already available in YouTube. So you can understand the exosome and the stem cell when you look at the video. There are many types of exosome technology, and many companies employ many strategies, and we are using adipose stem cell exosome. So when you look at the business portfolio of our company, we have mid-term and short-term business strategies. So we have the 2-track.
현재는 1번, 2번 비즈니스에서 저희는
And now
사업화를 해서
we are
전 세계에서
commercializing number 1 and number 2, and we are the only company that is getting profit out of this business around the world. And based on this experience, we would like to expand that to regenerative therapeutics or immuno-oncology. So these are the long-term plans. And we have Dr. Yang Latva, who discovered the RNA, and we are doing the RNA R&D as well.
앞으로 10년은 걸리는
Of course, this R&D will take about 10 years from now on.
저희가 추구하는 그 재생 에스테틱 외에
And
재생
along with the
또는
regenerative
다른 형태의
aesthetics,
바이오파마시티컬
there
파이프라인은
can be
보시는 바와
many
같습니다.
pipelines. So these pipelines are shown in the
잘
slide.
알려져 있고 단순한
This
편인데
mechanism of action is well known and very simple. Based on the anti-inflammation, we are focusing on skin
도마톨로지에 관련된
and dermatology
대해서
related diseases.
약과 사업을
We
추진하고자
are promoting the
그리고 비교적
business
최근에는
and drugs
줄기세포나
related to this.
줄기세포
And recently, stem cell
ARDS나
and stem
COVID-19
cell
치료제로 개발되고
exosome
있는데
are
특히 미국
developed
같은
as
경우는 한
COVID-19
회사가 US FDA
therapeutics.
페이지 3 어프로벌을 받아서
And
400명의
phase
환자를
3
대상으로
approval was given
COVID-19
to one
치료제를 개발하고
company,
있습니다. 상당히
and
빠르게
that
개발되고
company is developing the
아마도
COVID-19
그 회사가
therapeutics.
만약에
So
내년에
once
FDA
the
승인을 패스트 트랙으로
that
받는다면
company receives the approval from the FDA as a fast track, then I believe that will be the example of developing the COVID-19 therapeutics based on the stem cell exosome. About 5.5 years, we invested in R&D, and as I mentioned, we are focusing on the dermatology and skins, and we have more than 40 patents in Korea. And as you can see, there are many applications, and we are promoting commercialization and business. And I would like to specify several things. For example, skin irritation, hair blackening, and skin barrier reconstruction. These are the indications that cannot be handled with the conventional technologies. But so, by using a disposed stem cell exosome, we are trying to develop the drugs. And we already published more than 10 papers related to this. And in order to develop products, we need to make it injectables. So in the animal study, we used subcutaneous injection. and IV. So the key is capability to make injectables, and we are making a step-by-step preparation. This is the factory you can see where we can make the injectables. And about 3 years, we made an investment of $20 million, and we are completing our exosome project. As you can see, this is a 2-story building in Osong, and the total area is 21,000 square feet. And we have additional suite under construction to develop the stem cell exosome. So soon, injectable exosome clinical samples can be created, then we'll move on to the next phase. I believe you already know about exosome, so I will skip this. We are focusing on skin and dermatology. So when it comes to the adipose stem cell exosome, it is under the skin. So these stem cell exosome is one of the most important elements to maintain the healthy skin. So I believe in the future the interest will grow, and we focused on inflammation. So we are doing a lot of research and development for the inflammatory diseases, and anti-inflammation and skin barrier reconstruction and anti-itching are the indications we are focusing, and we are also trying to apply the patent. And as for the atopic dermatitis, using the adipose stem cell exosome, we measure the clinical score, and with consistency, we can observe the biomarkers and downregulate them. And in order to identify specific mechanism, we also conducted experiments how exosome can contribute to create normal stem cells. Eventually, skins are exposed to the external environment, and in order to prevent the viruses, here, the lipid barrier is very important. With aging, normal metabiology will be collapsed. And we tested the skin barrier to reconstruct them. And with the treatment of stem cell exosome, ceramide and key lipids were observed, and they were elevated. So in the
여러 가지
future,
그
it can be
피부의
applied to
재생이
wound
필요할
healing
경우에
and regeneration of
충분히
skins,
어떤 중요한 메커니즘으로 작용할 수
and
있다고
it will
생각하고
serve
있고
as an important
여기에서도 좀
mechanism.
일관성 있게
And
여러
here,
가지 프로 인플레멘터리
we
사이토카인이
were
저하가
able to
된다든가
see the
PSLP 같은
consistency of the data, such
중요한
as the
그
PSP
가려움증을 유발하는 분자들이
and the elements that
컨시스턴트하게
causes the
감소하는 것을
irritation of the
확인할 수
skin,
있었습니다.
and we were able to see that they
그리고
are
저희가 좀
decreasing
특별히 또 하나
consistently.
스터디를 한 것은
And we
엑소솜이
have
과연
another special
트랜스더멀
study
딜리버리가 잘 될 수
to
있을 것이냐
know whether it can be
그래서 저희가 일반적인
transdermal.
동물 실험을 하고
So we
특별히
conducted the animal test first,
엑소솜과 어떤 그 마이크로 커런트
and exosome was used.
엑소솜에 마이크로 커런트를 제공을 하면
And
상당히
once
많은
microcurrent
양의
of the
엑소솜이
exosome is
피부로 부화되는
provided,
것을 확인을 했습니다.
then we can
그런 이유에
confirm
저희가
that
실제로
lots of
케이스 스터디를
exosome
해보면
is
중증 아토피
transferred
환자인데
into the
당연히
dermatology.
이런
So
환자들은 스킨 베리어가 좀
for these
크랙이 돼
patients,
있고
we
아까
can see
말씀드린
that
어떤
skin barrier
메디컬
has cracks.
디바이스와 결합을 했을 때
And once we use these exosome combined with medical devices,
어떤 차별성을 가진
we
약물로
can
개발될
have
수 있는
the
가능성을
drugs that is differentiated from the conventional manner
이런
of
사이언티픽
drugs.
백그라운드를 통해서 저희는
Based on this
장기적으로
scientific background, in the long term, we would like to focus on the dermatitis and other indications. And when it comes to the adipose stem cell exosome, it is not well known in Korea. However, it is well known in the world, and it is growing. And when I participate in the dermatology conferences, with various technologies, we are trying to achieve anti-aging effect.
저희의 어떤 미션이나
And our mission or our technology
인텍트하고
is related to
그리고 노화가
have a normal skin conditions. And in order to have a normal skin condition, we first need to have the stem cell, and exosome will play an important role. As you know, there are various anti-aging technologies such as Botox or laser techniques, but eventually, in order to prevent the aging, we need to achieve the regeneration of the skin, and I believe it is closely related to skin diseases. And we are promoting the commercialization of this business by creating products. And we are applying this to the clinical conditions to have indication of wound healing.
그리고 또 다른 뭐 형태의 뭐 듀플맙은 잘 아실 겁니다.
And I believe you are well aware of the dupilumab.
이런 약물도
And this is an already available drug.
장기적으로 사용했을 때
But when we
특별한
use
사이드
these
이펙트가
drugs
나타나게 됩니다.
in the long term, there will be specific side
듀플맙
effects.
페이셜 레디니스라는
This is
명칭으로 지금
readiness
계속 사이드 이펙트가 나오고 있는데
in the face.
지금 파이프라인으로 보면 10개
So
이상의
when we look at the
모노클로널
pipeline,
안티바디들이 만들어지고
there are monoclonal antibodies,
일부분 사이드 이펙트를 가지게
and these will
이런
also
그
have
레디니스도
some side effect.
저희가 엑소좀
So,
지방교구세 백소점을
by using the adipose dispose stem cell exosome, by applying it in the topical area, we can also improve the side effect. And this person received the COVID-19 vaccine of Pfizer, and she developed the inflammatory diseases. So, by using the dermatology-related devices and our product, we were able to improve the condition. And our scalp is part of skin. So when it comes to the hair loss and scalp condition, we confirmed that adipose stem cell exosome can work. So MI-BRT7 and many other elements are included in the adipose stem cell exosome, and it is affecting to stem cell. So I'm showing some of the data we have. This is the hair follicle from a single donor, and we harvested this hair follicle and treated with exosome,
모발 성장이라든지
we were able to see the protection effect and growth of the hair.
연말 정도에
And
페이퍼를
at the end of this year, we plan to publish our paper. Based on this scientific data, we would like to commercialize the technology.
만들어서
So we are creating the products for scalp and hair.
80명의 환자를
As you can see, with 80 patients
미녹시딜을 같이
with minoxidil and other diseases have been applied, and depending on the concomitant use of the
다르게 나오는 것을 확인한
product we produced, the outcome was different. This is a Spanish dermatologist, and he is a PI in our clinical study, and he showed the three best cases. And in this slide, you can see the patient who used our drugs alone, and we are presenting this outcome.
앞서 말씀드린 내용은 지방줄기세포 엑소솜을
So I explained the adipose stem cell exosome and its commercial potential.
ARDS
Then, now I'd like to talk about the COVID-19 long hauler.
이미 그 스템셀 분야에서는
Already in the stem cell area, stem cell exosome
중요한 치료제가 될 수
is getting attention because it can become important cure for COVID-19. And many companies are developing it targeting COVID-19. Obviously, in our company, cytokine storm can be prevented, and proliferation of other cells and anti-opposites can be created with a stem cell. And our antiviral protein can be also confirmed. So in this area, we can see the publishment of the papers. So we did a pilot study. This is a H1N1 influenza infection model, and when we look at the survival rate, we improved the survival rate more than 45%. And when we look at other indicators, the graph is improving. So these are very consistent in inflammation model or cytokine model. We were able to show anti-inflammatory effects consistently. And we also did animal model test, and along with the H1N1 model, we were able to see similar outcome. So what I can say is that in the future, a disposable stem cell exosome will go into the clinical study, and already in the US, the phase 3 study has started, and we also submitted our paper. And there is another potential. The acute kidney injury does not have the drug. So in our perspective, kidney injury is very difficult disease to treat because there are three pathways. And the traditional way tries to prevent only one pathway. There are three pathways. However, if we prevent one pathway, then acute kidney failure will not be prevented. So in this area, when we use the stem cell or stem cell exosome, it will be the very effective alternative. We carried out multiple experiments, and 3 years ago, we also published a paper. And we were able to see the survival rate has been elevated according to the dose. And the body weight has been recovered to 80% of the normal ordinary group. So already there are development of stem cell or exosome, but our adipose stem cell exosome can be a good candidate for drugs. And for the sake of time, I will skip other data. And I would like to make the conclusion for the last 5.5 years in various areas. We carried out clinical study in vivo and in vitro study to seek out the potential for the business and commercialization. And of course, as a company, financial performance is very important.
세계적으로 현재 유일하게 엑소솜 사업
So our company has the only commercialized product, and we are generating revenue. For the last 5 years, every year we saw 50% of growth, and in the future, we believe that annual growth rate will be 100%. And in the first half of 2022, our sales was 118, and in the regenerative aesthetics, we are a global leader. And in the future, by acquiring another company, we would like to increase our sales to 100
뉴스를 드리자면 저희가
billion.
글로벌
And let me tell you another news.
월차들을 모아서 9월 17일, 18일 날
In September 17th and 18th, we would like to hold a summit called Aesthetic Exosome Summit. And
한국에 이제 LG
our partner Bennett and LG Chemical will participate as a host for this conference. And various clinical applications will be presented. Two key scientists and plastic scientists and GPs will participate in this conference.
감사합니다.
Thank you very much.
다음 발표 소개해드리겠습니다.
I would like to introduce the next speaker. The title is Exosomal Non-Coding RNA Therapeutics for CNS Disorder. And the presenter is the founder and the CEO and CSO of Ilias Biologics. And he received a doctorate degree from Yonsei Medical School. And he is also working at KAIST. And he published more than 130 papers in the international journal and has more than 30 patents. Please welcome him with a big round of applause. Thank you very much for the introduction, but there is one correction. I've been in KAIST for about 15 years, and I resigned from KAIST about 2 months ago, and now I'm dedicated to the company. And thank you very much for your valuable time for this exosome session. And I thought that how many people will turn out in this room, but I was very surprised that so many people are showing up. So thank you very much for your time. And I believe the order of the presentation is remarkable. The two previous presenters talked about the exosome. So they talked about the stem cell exosome, and they are trying to use the exosome as an avatar of the stem cell. But in our company, the previous speaker also talked about loading of the exosome. But in our company, we are trying to create a chicken that can lay the golden eggs. And two later presenters will talk about even more advanced and even more radical technologies. So there are so many technologies taking place in Korea. And exosome technology is still in infancy in the world. However, in Korea, we can see very active activities. And the two previous speakers talked about exosome briefly. It's been about 12 years. We realized that exosome can send the signals to the cells around it. So we realized that it is a very important communication route. So there are two meanings. First, the disease message can be read, and we can diagnose the disease early on. And the exosome itself can be used as a Trojan. So the drugs that can be difficult to deliver can be mounted on the exosome. And exosome itself also can be used as a drug or therapeutics. So our company is working on the latter. And how exosome can deliver the materials to the surrounding cells. As you can see in the slide, there is a target cell, and exosome, and they can be combined together just like the bubble of the soaps. And some will be degraded through lysosome, but part of them can be go through the membrane, and it can be going through the cell itself. So when you look at the core technology, actually, these are the technologies I developed in KAIST. And I didn't realize that doing business is very difficult. So I established my company 7 years ago. At the time, I developed Exosome. So it is like putting the elephant in the refrigerator. A big size protein drug can be loaded into exosome. I developed the technology. I will explain this later once again. But exosome will be delivered to the target cell by using the membrane, and it will be dumped in. So it should be protein-free. Then it can be effectively delivered to the target cell. So that was the beginning of the process. Eventually, we need to add something to exosome to enhance the therapeutic effect. That was the first target of our company. So when I published a paper, I named it as Explore. And now we are in the process of advancing the initial technology. And second, exosome is very interesting because most of the cells in our human body create exosome, but they have different content, and they all have different cells. So depending on the origination of the cells, they have specificity and tropism. Now the mechanism of action is now well known. However, a glycoprotein on the surface of the cells, such as adhesion protein, will be used, and it will determine the final target. So when we modify these, we can send these exosomes to specific cells, or we can avoid the delivery of the exosome. Many companies are engaged in this, and with our own proprietary technology, we are developing this technology. So improving the exosome itself is important, but delivery of it is also important. So when we combine these two technologies together, there will be synergy. And in the commercialization, what is important is increasing the exosome's efficacy and delivery. But at the same time, we need to produce it in a massive manner by scaling up. Otherwise, translation will be impossible. So for the last 7 years, we focused on these three areas. Without any of these, we cannot stand tall. The first core technology is, as I explained before, we need to have a large molecule protein. Of course, this is a difficult process. So aside from protein, we are also experimenting other materials. But when it comes to the protein, we need to be optically reversible. So depending on the light, it should be attached and disassembled. This is a non-scientific meeting, and it is already published. So I will skip this detailed explanation. But if you believe me, this optically reversible method is used. So when exosome is created from cell, we can load the protein selectively. And once it is mounted, then it will be attached to the membrane. And after purification, if we block the light, depending on the light, it can be attached and disassembled. So now we can have the exosome without the protein. So that is the core technology of our company. So first, we need to load the protein that is hard to load. Then after that, we also make the exosome as a free form. That we first created the proof of concept, and with lots of ways, we verified this. And cell-crystal recombination is used in our lab. So DNA recombination was used. we cut the specific sequence. This is kind of the immune system of the germs, and we use this in our lab. And this is called as Cree. And depending on the Cree, it will be activate, and cell will show the fluorescent signal. And by using the Cree, we can load the protein, and with the treatment, as you can see in the slide, we can compare this with naive exosome. In the naive exosome, there is no Cree. However, after the treatment, we can put the Cree, and it will move to the nucleus, and it will be moved to the nucleus, and there is a bags, outer membrane, and cyclone C release will take place, and the materials will be loaded to the exosome. So it is like a suicidal squad. So as I explained before, we have a cargo, and cargo should be loaded into the membrane. And we successfully confirmed this with our data. So we would like to know how big cargo can be loaded. 200 Dalton protein can be loaded, and the biggest protein we loaded is CRISPR-Cas9 gene editing enzyme. And other antibodies and antibodies, antigen recognizing single chain can be also loaded easily. So current antibody market is really big, but these antibodies were working outside of the cells because they cannot go through the membrane of the cells. But by using the exosome, we can deliver this into the cells. Then we can open up another big market, I believe. So this is another proof of concept of our technology. I explained the Cree, and we fusioned this to exosome membrane, and we also used various methods. And we also used IV, IP, and various administration route to deliver this to tissue, and we try to see in which tissue and cell Cree can be activated. And then we realized that kidney and brain, liver, they show the Cree repair activities. So with this Cree system, we can show the proof of concept, and we also can confirm the biodistribution of the exosome. So this is a very important tool. And the second one is about biodistribution and targeting. And including our company, there are many other companies. And without engineered exosome, IV or subcutaneous injection is required. Of course, there might be different peak time in the blood. However, 60 to 70% will go to liver, and the others will go to spleen. So they will go through the filter, and 5% will go to kidney. And interestingly, lung is a hypervascular, and the area is really wide, but only 1% will be delivered to lung. And when it comes to brain, even though many people are interested in brain, still the exosome can go through the blood-brain barrier, even though the size is big. I will show you the data. We loaded the Cree enzyme in the exosome, and we can see the distribution, but there are limitations. Once Cree is inside of the cells, then repair activity can be shown, but we cannot see how many Cree enzymes are inside of the cell. So, as a complementary method, we also use the repeat membrane and luminar side protein conjugation can take place. And we work together with the Bundang Seoul National University Medical Center, and PET imaging was used. And after an elapse of the time, then we analyze this biodistribution quantitatively and qualitatively. And biodistribution is important, but how we can change it is also important. So one of the technologies we secured is a CD9. So this is the surface of the exosome, and tetraspanning is there because there are four membranes. So N-terminus and C-terminus are heading to the inside of the cells. And there is loops, extracellular proteins, and other matrix interaction will take place in the second loop domain. And interestingly, there are two system bounds. So when we modulate this protein will not be created or localization will change. So in the past, we thought that we should not touch this. However, fortunately, in the near area, we confirmed that we can stably insert 100 amino acids, and these will be exposed well in the surface, and it is optimized for targeting. So that's why we call this as exotarget, and we also secured the patent. And when it comes to the blood-brain barrier, as I said, part of them will be delivered to brain, but it is only 0.5%. So it is not the satisfactory level. So in order to elevate this, we use the macromolecule. And one of the methods to go through the blood-brain barrier is the glucose is the main energy source, but it needs iron, cholesterol. So we use the protein that carry iron and cholesterol, such as the LDL, and it selectively uptail it. So we use a so-called hitchhiking. So we inserted the binding area, and compared to the knife, after 24 hours, other control group was washed out, but our exosome was retained. So the cargo that works in the brain can be used by using this method. Due to the limited time, I will skip this briefly, but when it comes to the pure exo, these are nanoparticle originated from cells. So we need to have a lot of cells, and we need to induce the cells to produce the exosome, and there will be upstream for culturing, and there will be downstream such as purification. And now we have 100-liter upstream process in our facility, and it goes through 15 cycles. And after 3 cycles in the CMO, we produce the exosome. And when you look at the in-process control, inside of the cell media, we understand that this is the exosome, but there are many contaminants. But after a series of purification processes, we finally have the pure exosome. This is how we secure the exosome, and now we are in the process of increasing and scaling up the 100-liter to 1,000-liter scale. And let me explain the pipeline. The first one is NF-kappa prevention, which is very important in the inflammatory diseases. When it comes to the NF-kappa-B, this is a transcript enzyme, and it prevents the delivery of the signal to the nucleus. So in our company, we harvest the NF-kappa-B, And even those inside of the nucleus, we take out from the nucleus. And in the cell level, we look at the exosome, and it can be applied to acute kidney injury model, and preterm birth model is established, and we had a joint study, and we already published this last year and a year before. And preterm birth will not happen when the low level of inflation is taking place. But with the inflammation, the fetus will not grow because the nutrients cannot be delivered to baby. So we can prevent the early birth. And based on this, we test toxicity. And this year in Australia, we will begin the first phase A study. And we also received the approval for the study. And we will begin the clinical study for the healthy volunteers. With this, I would like to conclude the presentation. Thank you very much. Thank you very much for the presentation. If there is any question, we would like to accommodate one question. If not, thank you very much.
And let's move on to the next presenter. And the title is Smart Drug Delivery Platform based on cell-derived vesicles, and which will be delivered by Dr. Oh Seung-woo, and he serves as a CSO from MD Immune, and also he serves as CEO for Biotherapeutics, and he also earned his PhD from University of Massachusetts, and also Dr. Oh has been leading the development of a clinical trial and project at Allen Institute. Please give him a big round of applause. Thank you very much. Good morning, ladies and gentlemen. As previous speakers are talking about the commercialization of exosome-based therapeutics, and it's my great honor to be here with these eminent leaders. And they talked about the reason why exosome is getting great attention, so that's also makes it easy for me to deliver my presentation. And exosome and vesicles are utilized in the talked in previous presentation. And we try to come up with new strategy to commercialize these exosome-based therapeutics. So actually, how can we commercialize those idea of exosome? And actually, that originated back to 10 years ago. And actually, my next speaker, Professor Ko from Postdoc, actually developed extrusion-based vesicle methods, and we got technology transfer. And in 2015, our moderator established our company, and we have employed more than 40 global employees and 13 research personnel, and our office is Seongsu-dong in Seoul. And in Seattle, we have global office focusing global BD. And CDV is a new technology, and GMP production process development are done by experienced veterans in exosome manufacturing. So exosome manufacturing pioneer Mario Zimona, doctor of and director of Berkeley Medical University, and we have enjoying joint partnership, thereby laying foundation for commercializing our products. So can skip this presentation because you're all aware of, and you are all aware of the reason why exosome come in spotlight. And the exosome researchers and in 2014 and 2015, the attempts to commercialize the exosome therapeutics have occurred. And in the early stage, many companies and research institutes think that, and the benefits of EV-based approach are natural messenger in cell-to-cell communication, and they are focusing on development of EV, but at the same time, they try to increase yield and manufacturing efficiency, and this is part that I would like to elaborate more. And so there is one way, extrusion-based method, and with this method, exosome potentials can be realized into a commercial product. And we are doing that exclusively. And actually for 2 to 3 years, US Kodiak, the UK Evox, and they are pioneers in the exosome field. And they have collaboration with large big pharma. And actually, they have concluded a deal which is amount to $1 trillion. Actually, this is not in clinical trial. This is in preclinical trial, and this large deal was agreed, which means this is a clear testament to how much attention to be maintained in the global big pharma companies. So now, let me take a deep dive into our core technology. As I mentioned before, one of the issues here in researching exosome is about exosome yield. So exosome can be naturally secreted by cells. So cell status, as cell type, and differentiation of cell, and the culture environment are factors that affect and decide exosome yield. And artificial adjustment has limitations. So going beyond that, so we think about how to easily obtain exosomes and vesicles from cell. So that got me develop extrusion technology, and the principles will be elaborated in this video. And cells goes through physically stressful environment, and conventionally we use membrane filters. So cells should go through a small hole, and there are a lot of pressure which leads to disrupt of cells naturally. And to our surprise, this broken down membrane naturally form a ball. So a variety of extrusions leads to miniaturized cell vesicles. So a lot of research support this, and these vesicles are similar to natural-driven exosome in terms of physical property and functionality, and CDV-based technologies are well known to this. And so we manufacture vesicle from extrusion, and cell engineering, vesicle engineering are used to load biological cargo and chemical cargo in this vesicle. At the same time, surface around it can be engineered in a variety manner so that we can engineer a more efficient vesicles that is not obtainable from naturally driven cell vesicle. And also in the early phase, 1 ml syringe-based extrusion was used. So it's a small syringe for your understanding. So we attach filter on the syringe, on top of the syringe, and we pull cell into this syringe, and it leads to extrusion of cells. And in the early stage of our establishment, we have been focused on how to scale up this at an industrial level. And with years-long researches in 2019, 100 milliliter cell suspension was intruded at once. We call this intermediate extrusion, and also intermediate scale extruder are allowing us to produce a larger scale of vesicles. And also, in order for these to be commercialized, there needs another step of scaling up. So last year, larger scale refined process and extrusion are research for that are under development. And now we are able to extrude 50 liter at once, and we have developed prototype for that. And to be commercialized, we have cooperation with manufacturers and other experts as well. So there are applications of our technology. For your understanding, there are two parts for here. So on the left side, and on the left side and this, you can see regenerative medicine. And there are a lot of cells that are very suitable for therapeutics, such as stem cells and other immune cells as well. And our vesicle technology ultimately breaks down cells, and we try to produce cell-like or miniaturized cells that are very similar to cells. And also, when the mother cell is vesicularized, those characteristics are inherited. CDV researchers and us are already proving this. So building on that, let's take stem cell as an example. So when we obtain vesicles from stem cells, it allows us to overcome the tissue barriers so that the therapeutic functionality of stem cell can be delivered to target region. And also, in terms of immune oncology areas are focusing on a lot of applications. On the right side, actually on the left side lies stem cells and immune cell, and it is the most weak point of primary cells are the difficulty of being engineered. So external genetics and materials injection are having limitation here. So in order for us to advance vesicles in terms of engineering, it also lies challenges. So to overcome these challenges, we have utilized a lot of methodologies to hack cell to produce vesicles, and produced vesicles are used to obtain vesicles with high functionality that we are not able to obtain from primary cells. And through this, we try to load cargo to be delivered to the target region. So we try to develop this as a smart drug delivery platform.
Let me tell you about the strengths of the vesicle in 3 ways. First, our technology itself is trying to overcome the limitations of the naturally oriented vesicles. So the first benefit is the scalability. I need to mention this. When we talk that this is scalable, of course, we have developed many processes for scalability, and already there are studies in the clinical phases. But when it comes to the stem cells, when we use the conventional purification process, we can obtain about 1,000 exosomes. However, from one cell, now we can obtain 30,000 CBB particles, and more than 200 times of yield gaps can be observed. So this differentiated productivity can lead to an easier operation of the process, and we can also reduce the cost. So we can create many benefits. And the second point is, as mentioned by previous speakers, this is oriented from human cells. And compared to synthetic vesicles, viruses, or cells, these vesicles we developed can be more safely delivered to human body. And another characteristics is the versatility. It can be viewed in many perspectives, but the first thing I would like to emphasize is that there are some cells that does not create exosome easily. In that case, even though we have good elements in the cells, but there are limitations in commercialization of the vesicles. But with the physical ways, we are making smaller vesicles. So it is not a natural way, rather it is based on the physical characteristics of the cells. So regardless of the cell culture condition, we can produce the vesicles. In this case, I believe we can expand this. And in exosome field, we are studying various technologies, and these technologies can be easily applied to our technology. So that's why I can say that our technology is very versatile. For the sake of time, I will only mention some of the data that can show the characteristics of the CDVs. When you look at this data, it is difficult to differentiate, but the blue one is a CDB and the red one is exosome. And the top one is exosome and the bottom one is the vesicles. So with extrusion, when we create the vesicle, physically it is difficult to tell the difference. And once the vesicle goes through enough extrusion, compared to the naturally made exosome, the physical characteristics and profiles are almost similar to the original cells. And in the middle, you can see the 3D structure by using the imaging technique called the SEM, and we can see that the particles are very consistent. And another characteristics is when we develop the CDV technology, eventually we want to deliver the drugs. And we would like to deliver the therapeutic molecule inside the cells to the cells. So the characteristics, of course, delivery should be considered first, and the cargoes should be released into the cells. But first, when we look at the uptake of the cells, we can observe repeated behavior. This is one of the data. We received the extruded vesicle and naturally obtained vesicle. And after purification, we used the vesicles. And from the extruded vesicle, we can see higher uptake in the cells. So these red dots are vesicles. And at the bottom, you can see the exosome. And we also used 4 types of cells. But we can observe the similar situation in other cells as well. And in the CDVs, the uptake was higher than naturally originated vesicles. And we also had an injection, and the efficiency was higher in the eyes. So a CDV can go through the retinal barrier, and it can also target many retina-related diseases. And stem cells and other materials are now being developed for a cure for retinal diseases. But using the vesicles, we can easily and effectively deliver the therapeutic effect. So this is a case where we can showcase the benefits of vesicles. And various mechanic studies have been carried out, and we have the clues that proteins make this difference. We created these vesicles, and in order to go to the next step, we need to have the GMP level manufacturing process. And for the last 2 years in Australia with the PMU, we collaborated with GMP facility. And as I explained before, we are using intermediate scale extruder to develop GMP compliant process, and it has been completed already. And documentation and process has been established. And the vesicle produced here will be used to understand the characteristics. And the stem cell CDV may have a lot of therapeutic effect, especially anti-inflammatory effect. So these CDVs are listed up here, and in various diseases, we can see the in vivo effect when it comes to the anti-inflammation. Due to the limited time, I will be brief. There are many ways to load the cargo, and these good technologies should be applied to our CDV. Of course, because the vesicles have the similar chemical and physical characteristics. So small RNA and messenger RNA, these large cargoes can be loaded in various ways so that we can see the effective outcome in the in vitro situation. And proteins are also being studied. Last but not least, in our vesicle, with additional engineering, now we can use the therapeutic antibodies, and we are targeting the tumor and to go through the BBB. In other words, we are now developing selective vesicles. We are developing we are not developing the therapeutic pipelines, but eventually we can support therapeutic pipelines. So we are developing good platform technologies, and through various partnership, in the future, we will support more pipelines. With this, I would like to conclude the presentation. Thank you very much. Thank you very much.
시간이 다 채워서
You spent all time, so we will move on to the next presentation right away.
차세대 면역 항암제
The next presentation is about scalable mass production of gram-negative bacterial outer membrane vesicles for the next generation cancer immunotherapeutic agents, which will be delivered by Professor Go Yong-sung from postdoc. And after BS and MS degree from Solange University, he got his PhD from University of North Carolina. And also, since 2000, he has 87 extracellular vesicle-related publications. And in 2009, he founded Korean Society for Extravesical Vesicles and Asia Pacific Society for Extracellular Vesicle in 2019. And now he serves as the president of that organization. And he founded Rosetta Exosome, serving as a CEO. So why don't we invite him to the stage? Thank you very much for your kind introduction. I am Professor Go from Postec, and I am also CEO of Logitech. And actually, I am asked to talk about clinical trial things. So we try to introduce next generation immunotherapy. But actually, before we take a deep dive into presentation, let me brief you on the company overview. And actually, our name tells you everything. We are Exosome company. And actually, I put an emphasis on our 3 platforms. So the first one is Exolute. This is a technology that is used to refine and mark exosome. And second platform is Ancolex, which is bacteria exosome, and third platform is Ghost nanovesicle. It is under development with MDimmune. The technology was originally developed by me, and we have achieved registered 20 patents and with 424 claims. So what I want to say here is that our patents, one patent has more than 20 claims, and we were established in 2016. And as introduced, for 22 years, I have been engaging in exosome field, and I have provided more than 100 articles. So lifelong experiences taught me a lot. There are a lot of difficulties and ups and downs as well. And 22 years ago, I started my exosome research, and at the time, I thought that after 2 years on, many people are all aware of exosome and they may pay attention to exosome. But unfortunately or fortunately, 10 years ago, this field is in limelight, which garners great attention from the world. And this reflects in the fact that in Korea, many companies are established in exosome field and they are doing business here. And the reason why people have great attention on exosome is that in developing new therapeutics, this is a new modality for them. However, what I want to put on emphasis here is that terminology. Many people are confused with the terminology. So, International Society for Extracellular Vascular, but the name is suggested by me, and many researchers accept it, and then it is used. However, the exosome is extracellular vesicle, and actually, exosome is secreted by the extravascular vesicle. So extracellular vesicle is not the common terminology. This is why we call this exosome. The reason why I tell you this is that academically important, all the cells have secreted extracellular vesicles. So it is evolutionarily maintained, which means it has a role to play. And over the 2 years experience, I've been taught that gram-positive can create exosome. This is one of the great achievements that I have made. This means all the cells create exosome, and it is evolutionarily maintained, and physiologically and physiologically, it has an important role. So, and when we do biocomposite analysis, we are able to do treatment and diagnosis as well. And second, over my experiences, I've been facing with many limitations. It is all about production and productivity. So I have come up with 2 solutions for productivity. First one is to use bacteria, and the other solution is to use mimetics. So I have carried out research on mimetics since 2010. With the technologies, MDimmune is doing their business. And during Dr. Oh's presentation, I looked up some data, and there were 600 citation for my article, which means many Korean people and other global researchers have carried out research and first generation vesicle. Not only MD, but also other Korean companies started their businesses out of this technology. So not only exosome, but also mimetic technologies are now in spotlight and limelight. On that note, our company has 3 platforms to show. So let me brief you on this platform. So I am a researcher, but I also have a lot of interest in businesses. In 2016, many people say that Spotify Science was established, and I was a scientific advice board at that time. And at that time, we have discussed the fact that how can we produce Exosome and do QC? And one of the participating member, interestingly,
분 회사에 소속했던 분들이 다 있었습니다.
was two gentlemen from
그다음에 세이키앙님 그리고 이제
companies.
누구죠? 오스트리아 지오마
Gioma was one of the participating member from Austria. So there were colleagues and we met with each other and discussed.
미국의 코디아 바이오
And building on that, the U.S. Codic Bioscience have carried out 3 clinical trials. But one of the important issues here is that how to mass produce in accordance with the CMC standard. So Codic Bioscience, as far as I know, have carried out 3 clinical trials, of which 2 have no issues in mass production because intraocular injection. This is the reason. The third is systemic injection. So Kodiak
자신들이 풀어야 될 문제라고
thinks and consider this as a challenge to be solved.
대량 생산이 있으면 되겠죠.
So mass production is of importance here, and
이렇게 생각을 해보면 많은 사람들이
many people, especially in the scientific paper or those in the business community,
주장하는 말을
argue
합니다.
that
대량 생산
we are able to mass produce,
세포가 내놓을 수 있는
but
exosome
the amount of exosome that can be secreted by the cells are limited. So the secreted exosomes can be obtained in some certain condition. This decides the demand, yield. So in terms of this obtainability,
얼마나 퓨어한
so we have to define the purity of my sample. So personally, we try to optimize isolation method,
순수함을 규명
which requires how to define the purity, and this. We have developed these technologies, and also we have a patent for this, and we also develop isolation technology building on QC. We have a lot of development of which
이 엑소점은 서피스 차지를 대부분 내거든요.
exosome shows negative surface charge.
저희 회사가 이제 등록된
So
특허 일이
So actually, we have a lot of patent. So positive ion-based physical chemical properties can be fully utilized to let us know how to refine exosome in a rapid manner. And also,
자세하게 설명을 드릴게요.
second platform is bacterial exosome-based innovative cancer immunotherapy technology. And third is about
개발하고 있는 기술 상당히 중요한 기술이고요.
ghost nanophysical, which is very important. We are developing.
많은 사람들
Not only Korea, but also in global companies have paid attention to this technology.
그 기술도 상당히 중요해요.
And for us, these technologies are important, but it has a room for improvement. So we come up with new technology, and the core concept is simple. So exogenous cargo should be mounted, which requires
엔도지너스 cargo를 버려야
we have to abandon endogenous cargo in lumen so that it can be filled. So how can we abandon endogenous cargo so that it can be replaced with desired cargo that I want to mount? So this is why we name it ghost nanovesicle. Of course, there are a lot of methodology, and we have shown proof of concept in terms of productivity, extrusion. The methodology is very similar to that of extrusion, and we have 100% higher productivity, and also we are able to confirm the lumen extracellular components are abandoned.
여러 가지 그 개념에서 저희가 이제
So out of a lot of concepts, we are able to prove this. But anti-inflammation medication are delivered in vivo test. Compared to the prednisone, we have witnessed 4 times higher rate. So this is very meaningful. Higher efficacy, so this is very meaningful. So anti-inflammation agent or anti-cancer immunotherapy, there are a lot of treatments, but overuse of this leads to adverse event. So increase in efficacy leads to little adverse event. So we have a core patent for ghost nanovesicle. and with surrounding patents as well.
그래서 이제 저희 회사는 그 박테리아
So bacterial exosome-based cancer immunotherapy is what we are focusing on, and also we try to utilize ghost nanophysical for a lot of tasks.
그래서 이제 그 원래 제가 이제 말씀
So I am supposed to talk about OncoLex platform, microbiome exosome-based innovative cancer immunotherapy. So I spent the rest of time to explain this technology and platform.
그 제가 이제 앞서 말씀
As I mentioned before, a lot of people know about the stem cell exosome. However, historically, before we doing research in this in 1960, we had carried out bacterial exosome research, and all the cells here secreted release exosome. So for 15 years ago, I have paid much attention to bacterial exosome, which is why how people and human can interact with environment, surrounding environment, and evolutionarily, bacteria non-self is something human try to defend. So with this, inflammation, immune system can be activated. This is what I thought.
근데 이제 그 아까 이제 조 대표님도
But CEO Cho and previous speakers mentioned that if we're successful in clinical trial in exosome, we are able to roll out therapeutics in exosome field. However, bacterial exosome therapeutics, Vexero, are rolled out in it was developed in 2013. It was approved in the US and EU, and GSK was a seller, and this is a meningitis vaccine. It used bacteria exosome. These revenues are expected to reach 1 trillion won. And so the bacterial exosomes are already therapeutics are already rolled out. So research on bacterial exosome taught me that mammalian exosome was also studied at research and its functionality as well. Bacterial exosome can activate host immune system with a small amount. So that got me thinking that bacterial exosome can activate immune response in tumor cell. So I think that the bacterial exosome-based innovative cancer therapy can be possible. And William Coley was very known for immunology, and he was a father of cancer immunotherapy. At the end of 19th century, he used bacteria to cure cancer. However, many doctors at that time were doubtful. So the Coley was outlier at that time. But a few years ago, Keytruda and innovative cancer immunotherapy developers were Nobel laureate, and the two were awarded William Coley Award years before awarding Nobel Prize. William Coley was a father of cancer immunotherapy, and also the U.S. Texas M.B. Anderson was a Nobel Physiology laureate, and he was a CEO of William Coley Institute at the time. Coley was an outlier at that time, and he tried to use bacteria to cure treatment, of which 1/3 of them shows response and 1/3 of them show no response, and 1/3 of them leads to death. At the time, they were not able to culture and QC bacteria. The concept of GMP, actually, this Mr. William Coley was giving birth to the concept of GMP in medical field. So we tried to use bacteria rhizome rather than bacteria. Then we can do works that William Coley want to do. And gram positive and gram negative create exosome, and we extract this, and we do test on mice test. So actually, this leads to complete regulation of cancers. Also, it prevent metastatic tumor growth. And surprisingly, when the tumor was completely regressed, actually we also see relapse or recurrence of tumor in mouse model after 1 month. But we should confirm that there might be our product are preventing the recurrence of tumor, and our mechanism of action is interferon gamma and MK T cell are dependent. So we established Roberta Exosome to carry out clinical trial, and we did mass production, as you're all aware of. Bacteria is easy to be cultured, so we have cultured 300 liters of bacteria. So we also put conditioned medium, and absolute technology is combined with this, which leads to development of mass production technology. Of course, there is no variation per batch. And with these technologies, we have carried out efficacy test. At the test level, we did mass production, and the functionality and efficacy were confirmed in this test as well. And one thing here is that we are able to turn cold tumor into a hot tumor. Of course, PD-L1, immune checkpoint inhibitor PD-L1, with the combination with this, we can increase significantly the efficacy. And with this mass-produced vesicle at a laboratory level, we have done multiple injection to test low toxicity, and also we have witnessed lower toxicity, and there is no effect on survival. So this mass-produced Rex 101 is in preclinical trial, and rat, beagle are model, and actually the toxicity of our REX 101 was leading to inflation in that model, but it's very manageable and recovered soon. So we are planning to attract Series B funding, after which we are planning to have a clinical trial. So actually, to put it simple, many Korean companies and researchers have been jumping into this market. So personally, it is very proud. But still, mass production and doing QC remains as a elusive and challenges. So we have long been engaging in development of this platform. So if you do have interest in this, we would like to look for collaboration partner and supporter, and REX 101 and other pipelines are development. So we are welcomed with co-development agreement. Thank you very much for your listening. Thank you for your presentation. If you do have a question, we will take one question from the floor if you have. Without that, why don't we move on to the panel discussion? So please wait until the arrangement on stage is made. So stay until the very end of this session while we are making arrangement on the stage.
네, 패널 토론 시작하기 전에 잠깐
Before we
안내
begin the panel
말씀드리겠습니다.
discussion,
오늘
I
엑소솜
have a housekeeping
세션에는
announcement. In this exosome
관심과
session,
보신 분들이 아무래도
we have
이제
many
엑소솜
participants who are
관련된
interested in the
연구도 하시고
exosome,
계실 것
and I
같고,
believe you are
또
a
앞으로
researcher,
연구에 관심
and
있으신
in
분들이
the future,
오셨으리라
you will
생각해서
also do
저희
the
엑소솜
exosome research.
비오플이
So
모인
the
그
exosome
협의체가 하나
companies
있거든요.
created
작년에
an
엑소솜 산업
consulting body, the Association of Exosome Companies.
지금 엑소솜
We created that last year, and
30개 기업이
about 30 companies are the members of the
돼
association.
있고, 그래서 상당히 많은 이
And
안에서
inside the association, among the exosome companies in the future, we will facilitate the networking.
메인으로 이제
So
연구하지 않으시고
even if you,
제약사에서
exosome is not the focus area of your research, but still, if you are interested in exosome, you can become the member of this association. Then you can have a chance to participate in the networking, and you can get the information, and we'll also provide information about future events. So if you are interested in the association, please contact us. Then we'll begin the panel discussion right away.
먼저 좀 준비해 온 그 질문들이 있는데
We have prepared
오늘 발표해 주신
questions,
분들의 의견들
and
듣고
we would like to listen to the opinion of the presenters. And if there is any questions from the floor, we'd like to accommodate the questions. Please deliver the question, then we will cover
시간이
that.
많지 않아서
We don't have much time,
제가 하는 질문에
so
답변은 의견은 좀 간단하게 이렇게
please be brief when you answer the question.
질문은
Let me give you the first question. In development of the exosome therapeutics, the most frequently asked question is heterogeneity. And exosome CMC is very difficult according to many people. So how can we overcome this problem? And how do you think we are overcoming this problem? In regard to this question, please give your opinion. We have a microphone. Please be casual. I will go first.
줄구석 엑소솜에 일단
We are developing the stem cell exosome,
까지 뭐 많은 연구자들
and we have been doing a lot of research with our researchers.
줄구석부 같은 경우는
And when it comes to the stem cell, we believe that there is no problem in CMC because the stem cell itself
많은 임상을 통해서
has been going through many clinical studies. So many toxicity issues have been addressed. And
아까 과영 교수님께서 데이터를
as Dr. Park said, there are consistency in the data.
양에 대해서는
But when it comes to the production mass, we need to go through the immortalization.
CMC가 어려워서
But CMC is not the problem for doing the business in stem cell exosome. I don't think so.
아까도 말씀드렸지만 이미
So as I mentioned before, in the US FDA, a stem cell exosome is used for the clinical study of phase 3 for 400 patients. And this clinical study has been approved. So at the end of this year, the clinical study will be completed. So as a member of the stem cell exosome field, I have high expectation. Regarding this question,
20년 이상 연구해 오신 우리
we have Mr. Ko Yong-song, who studied this for more than 20 years. You studied the stem cell and various other areas, so CMC and heterogeneity can be answered by you.
첫 번째는
First,
저의 원래 전공은 화학이에요.
my original major is chemistry, and I purified protein when I was in university. And recently, I received these questions from many people. So I cannot understand,
화학자 입장에서
because as a chemist, purification of the exosome is even easier than purification of the protein because the size is big and there are many methods. So once you understand the property, then the purification of the exosome is even more easier.
그런데 이제 남아 있는 문제는
And then the remaining problem is heterogeneity.
역으로 질문.
But I'd like to ask a question rather.
저희 국제학회 사람들
In the international conference, I had a debate and I raised this example.
여기 계신 모든 분이 그냥 MSC가 내놓은 엑소좀이라고 한 거예요.
Let's say that all of you are the exosome of MSc and Korea is MSc. and the Netherlands people are cancer cells, but we are all different. But from the foreigners' perspective, we look same. So this concept is very important. So when you cultivate the stem cell, all stem cells are different, even though they are similar.
나오는 엑소좀도 당연히
So the exosome secreted from these stem cells will be also different. But the remaining problem is homogeneity or heterogeneity. Whether we will have a strict line between homogeneity and heterogeneity. But everything is relative characteristics. So there is no homogeneity in absolute terms, but the range is different. So from the CMC perspective, we need to set the right range. Then regulators will accept the outcome. So when we create the tennis ball, we have the diameter and there is a margin of error. Just like that, we can convince the regulators in the exosome. I believe the EOSFD has the same position. Thank you very much for your answer. I can understand your experience. Thank you very much. We'll move on to the next question.
So globally, the development of exosome is an area of business startups. And in Korea, there are a lot of companies specialized on development of exosome therapeutics. The amounts to more than 30. And actually, this is a very vibrant environment. So the level of domestic exosome technology can be matched with pioneering companies. So what is your take on this? What is Korean companies' technological level and ranking in this regard? And I would like to add, actually, Cha Chul-hee, the CEO, he's doing global clinical trials. So could you please answer this? So actually, as a disclaimer, I am not blaming or ignore global companies. But actually, Korea's exosome development is robust because it is all attributable to the greater role of Professor Ko. And actually, Korea is exposed to this area, and we are on the same equal starting line. And at the same time, with regards to stem cell industry, Korea MFDS is active in supporting this industry. And also, there are some quantitative measurements to compare. However, when we do look at it as a quantitative manner, and qualitative manner, when we look at this in a qualitative manner, the two leading global companies are Boston Kodiak and the London-based Evox. There are two trailblazers in this regard, and they were established in 2016 both, and they got funded aggressive manner, and actually they got more than a lot of investment more than us. Of course, the Kodiak have carried out 3 clinical trials, of which cutaneous cancer injection. For us, actually, we do systemic injection, and other team tries to do systemic injection for stroke patients. And this is an uncharted path for global 2 companies. So I have visited international conferences and I have listened to many presentations, and we have shared the robust data with them. But we are able to remain competitive in this field. So actually, you are so leaning toward the Korean companies, right? So biomedical technologies in Korea are advancing rapidly, but at a global level, we have a long way to go, right, to be on par with the level of the global pharma companies. But in terms of pharma company, it is very newly born technology, but we are able to be on par with the global pharma, and this is one of the advantages that we have in exosome industry. Thank you for your kind answer. And after listening to your explanation, exosome companies in Korea can be cheerful. And let me throw another question.
The topic of this session is limitless expandability of exosome, and this is a highly expected therapeutic platform for many indications. So, what would be the most highly potential indications, and what are the basis for that expectation? Of course, different companies have different areas for development, so there are many things you would like to say. So, in which area exosome can be successful early on, and which one has the highest potential, do you think? Professor Bang? I would like to talk about the neurology. I'm a professor treating neurologic diseases. And many people ask me, why are you in the neurology? Because there are many failures. So some people are resistant about neurology because there are many failures. And the clinical practitioners would like to overcome the neurology hurdles, such as stroke and Alzheimer's disease. We had the failure, and we realized that this is not just for the one target. We need to target multiple indications. That is the consensus. So in this regard, when it comes to the drugs, in terms of the neurology, we had many experiences before. So as I mentioned before, we need to have accurate profiling, and we also need to take approach to target multiple things. And as other speakers mentioned, in cancer and in other area of medicine, the targets can be different and effectiveness can be different. So I believe their approach can be totally different from us. So as you said, depending on the target and depending on the company, the approach can be all different. So neurology diseases can be the target for exosome, and I believe exosome has the potential. So is there any other opinion? I would like to add my comment. Exosome has many benefits, and one of them is permeability. So I can go through the barriers, biological barriers, and that's why the central neural CNS is one of the targets. So such as turning cold tumor to hot tumor, and we have the profile. So placenta is difficult to go through because it protects the fetus. But the umbilical cord can be a pathway. So it can be a good way or a bad way. But if we interpret this in a good way, then we have another tool for a fetal compartment. And when we think about the market, CNS and cancer would be the big market. And with exosome, just like Exosome, we can use the topical therapeutics because it can be easily applied. Then it can be also applied to anti-inflammatory indications. The market itself can be big or small, but anyways, these areas will be realized first. Then it will be expanded to other unmet needs. When I look at overseas companies and overseas researchers, I can summarize that many Korean people create a company and they have interested in this area, but it is a similar situation in other countries. So if you are operating your company or if you want to create your company, I would like to give you the advisor. You want to be the first penguin, so you want to be the first mover in the big markets such as cancer or CNS. However, in the other countries, they have good human resources and good funding. Thinking about that, creating a success story is very important. So I think we need to take the approach based on common sense. So there should be pipelines. One of the pipelines should be targeting the first mover, but you also need to have the pipelines that can target the rare diseases.
네, 감사합니다.
Thank you very much. We don't have much time.
저희가 이제 질문이 한두 개 더 있는데
We have one or two more questions prepared. However, if there is any questions in the floor, we would like to take them. Please deliver the microphone.
여기 요 앞에 예, 마이크.
And I can locate him, so could you please give him a microphone? So I'm from Jina Innovation, and now it's very hard time for funding, right? But new biopharma company in Korea have lack of competitiveness because of lack of global funding. So many companies have technological transfer, but there's no global funding or lack of global funding. So we have to attract global funding in competitive area. That's why I believe Exosome area is of great potential. We have done a lot of competitive works in this field. So we need to invite global VC to put on a lot of investment in Korean companies. They can be a good success story, but we have a dilemma here. We have a highly competent technology. However, global VCs are not willing to make an investment in here. This is because Korean companies, governments, or there's a weak point in global document CMC part in Korea. So we have to put a lot of attention in this weakness point so that we can lay a foundation for attracting global investors to the Korean company. There can be a faster pathway. Thank you very much. So is there anyone who wants to have a comment here? Yeah, that's a great idea. And one thing to add on is that when we get funding from the global VC, Korean-based companies are not eligible for that, from my perspective, because there should be a robust data to support it. But technology itself can be homegrown in Korea. But in terms of the US cases, actually, VC is not willing to make an investment if a company is coming from different state, because there are enough companies that share the same location with the VC. So the global VCs might know well Korean code of commerce. However, if that's not the case, the hurdle is very high. And I would like to add on this. Recently, I have been contacted by European VC, and they searched for our company. What I thought that is strong IP portfolio is great important. So as you mentioned, documentation and the governance are also playing an important role here, right? As you mentioned before. So actually, in order for Korea to culture great company, we have to build a strong IP portfolio, and we have to support them. And it have to do with new modality. This is not for patent for use, but this should be fundamental. And this is a prerequisite for us to attract VC companies' investment and attract attention as well. 마이크를 켜셔야 통역이 제공됩니다. 마이크를 사용해 주십시오.
As a matter of fact, Korean bio companies need to be globalized, and I believe you suggested a good direction. And we have another question. My name is Jung Han-chul. I'm sorry that as a venture capitalist, I have high interest in the Exosome. And this is very privileges for me to meet all of you. And thank you very much for your insight. When I look at the exosome, I thought that it is like DDS, so it is just a delivery mechanism. However, recently, many VCs are focusing on gene therapy, and exosome is a good way to deliver the genes. It is 100% better than other method. And you also mentioned mRNA. And in gene therapy, what would be the direction for the future? I believe you are studying about the loading, and it will be loaded and delivered, and it will be expressed. So the FDA will ask how much expression can we observe. So what would be the first? So can you provide the insight? I believe we need to have a lot of discussion, but due to the limited time, the organizer said that we need to wrap up the session. So is there anyone who would like to give the answer about gene delivery? If I be brief, everyone can make argument, but when it comes to the mRNA vaccine, Moderna has never been successful in mRNA, but it was successful in COVID-19. So with a small antigen, vaccine can be created, but when it comes to the therapy, depending on the indications, target indication, delivery efficacy can be all different. So there is no one absolute answer for that. So this is related to the efficacy. I think this can be the answer for your question. It is very shameful that we cannot cover all the questions, but thank you very much for all the turnouts. And I hope that the exosome industry develop in the future. With this, we would like to conclude the session. Thank you very much.
수고하셨습니다.